Hey everyone, it's Dr. Marcon. This is Lab 32, Control of Breathing. This is our last lab of the semester. Yay! So this is actually a fairly simple lab that you could actually perform at home. However, I do have to say, if you are going to hold your breath, please don't pass out. We don't need that. So this lab is called ventilatory control. So we're basically having groups, actually every individual is going to hold their breath, and then we're going to take breath holding times. So we'll measure the breath holding time under different conditions. And what we would normally do is everyone would do this individually, and then we would get a class average of breath holding times. So we know that the respiratory system is highly regulated to maintain arterial blood, carbon dioxide, oxygen, and pH at nearly constant levels. So any change in these levels is detected by chemoreceptors, which will then send a message to the brain to alter breathing to maintain that normal level. Now, in this experiment, we'll examine the effects of altering the initial status of blood gases and or the volume of air in the lungs on the breath holding time, how this affects how long we can hold our breath. So based on the observable data from both an individual and classroom measurements, we can then determine whether the primary driving force for ventilation or for breathing is driven by primarily by either a lack of oxygen or an increase or buildup of carbon dioxide. Okay. So again, the objectives include to examine the effects of altering the following on breath holding: volume of air, how much air we put into our lungs or take out of our lungs, as well as the initial status of blood gases, primarily oxygen and carbon dioxide. And then determine whether it's a lack of oxygen or an increase in carbon dioxide, which is the primary force or the stronger driver of breathing. Now again, just some general notes. The individual will hold their breath until about the same level of discomfort each time. Now, this is not a competition. There's no cheating. Hold your nose if you have to. And please, no passing out. We don't need people passing out at home. So we will then determine breath holding time under these 6 conditions. So first, taking a max inhalation, so breathing in as much as you can, and then holding and measuring breath holding time. The next condition is just a normal inhale and then hold. Then we have a maxed force exhale, and then you hold your breath. And then you have taking deep breaths or hyperventilating and then holding your breath. Rebreathing, so basically rebreathing either a plastic or paper bag and then holding your breath. And then exercise for a little bit and then holding your breath. Now, in our lab, we would not have people doing exercise within the lab. We would tell students to go outside, do a bit of exercise, and then measure how long they can hold their breath. So we cannot do in class. From what I was told from the other instructors, it basically sounds like a herd of elephants or even an earthquake, which would freak out the other classes. So that is why this part of the lab, the exercise portion, has to be done outside. So the first 3 conditions, we have to assume that with procedures A through C, the starting lung volumes will vary. So we're going to be changing the lung volumes for A through C. However, the blood gases, oxygen usage, as well as carbon dioxide production, as well as pH are going to remain constant. So for the first condition, normal inhalation and then holding your breath, this is to get our baseline. So again, we just inhale normally and then hold your breath. And then time it. For procedure B, max inhalation, you breathe in as much as you can, and then you hold your breath. With condition C, you have a max forced exhalation, and then hold your breath. So what we expect to see is with max inhalation, we are increasing lung volume. And if you look at the numbers, so our normal inhalation baseline for breath holding for an individual, this is this column right here, and this is the class average. We saw that with normal inhalation, the baseline was about 36. This, I believe, is a normal, apparently healthy 21-year-old female. You can find this information in your lab supplement. So this individual, the breath holding time was 36 seconds for just normal inhalation. This is her baseline. And the class average was about 41. With max inhalation, we saw that the breath holding time increased slightly with this individual. So they went to 53. And the class average also increased. So we can see that the class average was about 58 seconds. Okay, so we see that with increased lung volume, we are able to increase the breath holding time. With max forced exhalation, you can see in this individual, it has decreased from her baseline. So it went from 36 to 21. And we can see that the class average also decreased. So based on this results, we can see that with a decreased lung volume, we decreased breath holding time. So max inhalation had the longest breath holding time because it had the max amount of air in the lungs, or max amount of lung volume. So what can we conclude from this? We can see that hold time is directly proportionate to lung volume. So as lung volume increase, breath holding time also increases. And the opposite is also true. As volume in the lung decreases, breath holding time also decreases. The more air in our lungs, the longer the breath holding time. So the more oxygen in our lungs allows you to hold your breath longer. Whereas the smaller lung volume, there is less space for carbon dioxide to go into. At this point, carbon dioxide builds up quicker with a smaller lung volume. So increase lung volume changes two things, increase in the amount of oxygen and decrease in the amount of carbon dioxide. So a basic increase in dispersal. However, it doesn't really, these three procedures of this part of the lab doesn't really answer our question yet as to which is a stronger driver of ventilation or of breathing. Is it oxygen, an increase, or I'm sorry, is it a decrease in oxygen or an increase in carbon dioxide? And that's what we're going to take a look at in the next part of the lab with procedures D, E, and F. So in procedures D, E, and F, we're going to be under the assumption that the starting lung volume is going to be constant, whereas the blood gases and pH are going to vary. So that means oxygen usage as well as carbon dioxide production and pH levels are going to vary or change. So the first condition we're going to take a look at in this section of the lab with D is undergoing deep breaths. So deep breaths gets rid of carbon dioxide, so the partial pressure of carbon dioxide goes down, whereas arterial oxygen will stay the same. So we can see from our table with this condition that breath holding time in the individual actually goes up, and it also goes up in the class average. So result of deep breaths is an increase in hold time. Now what can we get from this? Our original question was, what's a stronger primary motivator for ventilation? Is it a decrease in oxygen, or an increase in carbon dioxide? If we look at this, if we think that oxygen is the primary motivator, we see that oxygen stays the same. So if oxygen is the primary motivator, then you would think that breath holding time would also stay the same, or also shouldn't change. But we do see that if carbon dioxide would be the primary motivator, and if carbon dioxide changes ventilation. So for example, we see that carbon dioxide went down, and we see that there was a change. We have an increase in breath holding time. So this is the first condition that shows that it's consistent with the idea that carbon dioxide is a stronger catalyst for ventilation. And then we take a look at condition E, rebreathing. So rebreathing air in a bag, and then measuring the breath holding time. So we allow the individual to breathe in the air bag and then take a breath, take a maximum breath in from the bag. We know that, in normal ambient environmental air, carbon dioxide is a very small percentage of the gases in the regular environment. However, if we breathe into a bag, a paper or plastic bag for, a few breaths, we increase the carbon dioxide concentration that the individual is breathing in from the bag. So what are results? We see that with rebreathing, the breath holding time in the individual actually decreased. And you can see that also in the class average. So we know that arterial carbon dioxide increased, arterial oxygen stayed the same, and this resulted in a decrease in hold time. So we see that the breath holding time, when the individual was able to breathe more carbon dioxide, actually decreased. So again, if we think that the oxygen is the main driver of ventilation and oxygen didn't change, if we actually hook someone up to a pulse oximeter, we know that their oxygen saturation will be about 99%. If the oxygen stayed the same, then the breath holding time should also remain the same. But we see that that didn't happen. What actually happened was carbon dioxide, arterial carbon dioxide increased, and this caused a decrease in breath holding time. So again, another condition that kind of supports the idea that carbon dioxide is the main primary driver for ventilation. Now with exercise, we kind of saw this in the beginning of the semester. With exercise, the individual, increases their oxygen consumption through an increase in ventilation, also has an increase in carbon dioxide production. Now, we can see that arterial carbon dioxide, arterial oxygen, and pH levels basically stayed the same because of an increased ventilation. However, we did see a decrease in breath holding time. And it's because that we were not able to remove the increased amount of carbon dioxide that was produced by exercise. So again, increasing carbon dioxide levels basically decreasing the breath holding time. And we can actually see that in our table with the individual having, a breath holding time of about 8 seconds and the class average being about 11 seconds. So what is our conclusion? Our conclusion is that carbon dioxide is a stronger driver of breathing and ventilation compared to oxygen. And we can see that evidence in conditions D and E. We can see that carbon dioxide is inversely proportional to breath holding time, meaning if we have an increase in carbon dioxide, we have a decrease in breath holding time. Another thing that we can conclude is from the earlier part of this lab is that as we increase lung volumes, we can also increase breath holding times. But the takeaway from this is that carbon dioxide is the gas that drives ventilation. Now, oxygen does play a role, but and usually it's when levels get really low, but it's such a minimal role that we have people that can actually pass out if they hold their breath for too long. The oxygen sensors in the body do not increase the drive to breathe. As an example, people can drown while holding their breath underwater, or they can, again, pass out by continuing to hold their breath. So it is changes in arterial carbon dioxide that will affect breath holding times, because carbon dioxide is a stronger driver of breathing and ventilation than oxygen. Okay, and that concludes Lab 32, Control of Breathing. There aren't any worksheets for this lab. This is something that you can actually try at home and test on your own, but make sure you know the concepts of the primary forces that drive ventilation and how each of these 6 conditions affect breath holding time.